Thanks to Leo for the introduction. I'm very happy to have this chance to share our team's recent work, ProTrek, and I think we can start now. So we know understanding the relationship of protein sequence, structure, and function is very important. And nowadays, we have many tools for this analysis, such as BLAST or MMseqs2. These are sequence-based alignment tools. Or we have Foldseek, TM-Line, and these are structure-based alignment tools. However, these alignment-based tools always retrieve candidates based on sequence or structure similarity, filtering out dissimilar proteins. But from this figure, we can see dissimilar proteins often can share similar functions. For example, in this figure D, you can see the x-axis is sequence identity and the y-axis is the functional similarity. So for protein pairs that have low sequence identity, their functional similarity can be low or high. And from the figure A, we can see first the part 1. So the part 1, the protein pairs have low structural similarity, but they have high functional similarity. And from the part 4, the protein pairs have high structural similarity, but they have low functional similarity. So this figure shows that these similar proteins can often share similar functions. So if we want to use alignment-based tools to find some proteins like remote homologous proteins, they always struggle to do that because their strategy is to filter out those proteins with low sequence or structural similarity. So we think a feasible way to solve this problem is to incorporate protein function description into our pre-training process. Previous approaches, some approaches model protein functions as discrete labels such as EC and geo numbers, but this way to incorporate protein function always limit the model's usage. For example, we cannot retrieval proteins given a human language description, or if we have a desired function, we cannot retrieve proteins using these models. Besides, some other models use the parts of the descriptions in the UniProt web page to incorporate protein function. For example, ProtST or protein DT, or in this year we have many these cross-modality models like protein clip or some other baselines. So, but these models only use parts of descriptions in the UniProt web page. We know UniProt web page contains many abundant protein data information. For example, protein names, protein function, or subcellular locations. Many subsections contain different protein properties. However, these models only use parts of descriptions such as protein names or protein function or protein similarities. So we think this will limit the model's usage and will prevent them from acquiring a more comprehensive understanding of protein function. So to solve this problem, we list all information contained in UniProt web page. The UniProt page contains over 50 types of subsections containing different text information. So here we split them into 2 types, i.e. the sequence level subsections and the residue level subsections. So what are sequence level subsections? For example, here we choose the function subsection. You can see, so in UniProt web page, the function subsection contains several complete sentences to describe a protein's property. So this sentence can be directly used for pre-training, such as contrastive learning, because it's a complete sentence. However, for residue level subsections, here we use the mutagenesis for for example. So it is not a complete sentence, but just it's just a table. And from this table, you can see the mutation information, the original amino acid, the mutated amino acid, and the position, and a description about this mutation. So from this table, you can see what will happen when this mutation happens. However, you cannot input this table to model because it cannot understand such format. So this is the residual level. This is why we say them as residual level, because it's not about the protein's property, but several amino acids in this protein, and it cannot be directly used in the model. We have to reformalize it into a complete sentence, and then we can feed them into the model for pre-training. So to solve this, we use the ChatGPT to generate several sentence templates. For example, here we have a cofactor Fe2, and we use ChatGPT to generate. We input ChatGPT a basic template such as a cofactor of this protein is something. And then we ask ChatGPT to generate several diverse sequence templates. For example, something serves as a cofactor for this protein, or this protein utilizes something as a cofactor. So then we can just fill the keywords into these templates. For example, the Fe serves as a cofactor for this protein, or this protein utilizes Fe as a cofactor. So to do so, we can generate sequence templates for each of these subsections, and then we can reformalize this tables into a readable complete text. And this text can be then combined with sequence level data and fed into model for training. So to construct our dataset, we use the SwissPro database and UniProt 50 database. The SwissPro database contains around 500,000 proteins, and they are human reviewed, they are reliable, and the annotations in the UniProt 50 proteins, they're machine annotated and they're not reliable. So our strategy is we first construct 15 million SwissPro samples, and we use these samples to train the first version of ProTrek, and then we use ProTrek to filter out the unreliable samples from UniProt 50 samples. And finally, we got 25 million relatively high quality UniProt 50 samples. So combine these two type of samples, we constructed 14 million samples. And each sample is a sequence, structure, and function tuple for contrastive learning. You can say the text information from UniProt webpage contains over 50 types of descriptions compared to previous baselines that only contained 3 to 4 types of descriptions. So here is our ProTrek model architecture. ProTrek contains 3 modality encoder, sequence encoder, structure encoder, and text encoder. During training, we encode proteins into sequence and Foldseek sequence. So here the structure encoder is a BERT model, and the input sequence is a 3D sequence encoded by Foldseek. So during training, we encode the protein into sequence and structure, and we also input its corresponding function. This text description can be function or geo annotation or subcellular localization. So we include 6 contrastive learning loss functions. You can see for sequence and structure, we have 2 loss functions. For structure and text, we have 2 loss functions, and also for text and sequence. So here are 6 contrastive learning loss functions. And also we want to learn the relationship in the single sequence for both sequence or structural sequence. So we added 2 additional masked language modeling loss functions for sequence encoder and the structure encoder. During training, sequence and the 3DI sequence were randomly masked at 15%, and the model are expected to recover these masked tokens. So here are the total loss functions we adopted during our pre-training. 6 contrastive learning functions plus 2 masked language modeling loss functions. And after pre-training, now you can do the flexible retrieval from any modality to any modality. For example, if you have a sequence and you want to know what is this, if I have a protein sequence and you want to know what is this protein's property, you have a text database, and now you can just encode this protein into a embedding point, and you can search. You can search its the most the nearest labels for its predicted function. Or if you have a desired function and you want to search some proteins that share this function, that have this function, you could encode these tags into an embedding, and you construct a sequence database. And by finding the nearest sequence points, you can find some proteins that have this function. Or if you have a structure and you don't want to do cross-model retrieval, and you just given a structure, you just want to search the similar structure, you can also encode a protein structure into a point and adopt a structure database. And by doing so, you can do the structure to structure retrieval. So it's really flexible. In all, you can ProTrek supports 9 retrieval tasks. So from these 3 modalities to the 3 modalities, so 3 times 3. So we did a lot of experiments to test the ProTrek's retrieval performance. So the first is the protein text retrieval. So for each subsection in UniProt, we test its retrieval performance. So for both the residual level retrieval and the sequence level retrieval and their mixture. You can see, compared to previous baseline, i.e. the ProST and the protein DT, they use the descriptions they also used the descriptions in UniProt to train their model. However, they only include 3 to 4 subsections, such as protein functions, protein names, or protein similarity. So they did not include so many subsections for their pre-training, and you can see their performance are worse. And in most subsections, their retrieval accuracy close to 0. So in comparison, ProTrek, both sequence encoder and structure encoder, they achieve relatively high score, especially for sequence level retrieval. So because we know the sequence level subsections describe a protein's property, a whole protein's property, and the residual level retrieval only are specific to some amino acids. So in some residual level retrieval, ProTrek may perform not that well, but for sequence level retrieval, it can be very accurate. And we also validate it using some new released literature. So we use some new literature, their protein, we predict their function, and we found the predicted function corresponds to the function described in their literature, So as we mentioned at first, those alignment-based tools cannot find dissimilar proteins that share the same function. We test ProTrek with this thing, and we find that ProTrek can find proteins that are not structurally similar but share the same function. Here we use the geoterm as an example. For example, we choose the zinc ion binding. This geoterm its natural text description is proteins with the molecular function zinc ion binding. And in our test set, it has 220 proteins that have this function. And we choose one of the proteins P13243 as query template to be input to Foldseek. And Foldseek will search candidates in our test set. Our test set contains around 4,000 proteins. These proteins are not seen during our pre-training. And we use this test set to construct a database for Foldseek to search. However, we found that for Foldseek, when we use this template as a query template, Foldseek, the top candidates found by proteins, only 18 of them are true, and the remaining 200 candidates are false. However, if we use this text description to input to ProTrek and we do text to protein retrieval, you can find that 198 hits are true and only 22 hits are false. So this shows that ProTrek can find some proteins that cannot be found by Foldseek. So to better illustrate this, we visualize some proteins found by Foldseek and ProTrek. You can see here we list those proteins and we calculate their TM scores. So here, this is the query template, and these two proteins were found by Foldseek, and these three proteins colored by blue were found by ProTrek. So you can see the proteins found by ProTrek, they have a relatively high TM scores. However, this protein, for example, is not true. So this protein does not have this function zinc ion binding. However, Foldseek retrieve it because it has high TM score with the query template. However, for ProTrek, you can see the TM score among ProTrek's proteins and with the query template, their TM scores are low, but they share the same function, zinc ion binding. So that is why we incorporate protein function during our pre-training. By directly modeling protein function, we ProTrek search proteins not only based on sequence or structural similarity, but also based on function similarity. So additionally, we test this feature with more experiments. For example, we use ProTrek sequence to sequence, structure to structure, and sequence structure sequence retrieval compared with other baselines like Foldseek, BLASTP, MMseqs2, or Diamond. We follow this experiment. So for each protein in the test set, we search this protein against the test database, and we count the number of true hits, and also we calculate the distributions of TM scores. So you can find for all models and tools, they can find proteins that have high TM scores. So you can see this part. So all proteins that have high TM scores, they can be found by ProTrek or Foldseek or these alignment-based tools because they find proteins based on sequence or structure similarity. But for proteins that have low TM scores, these alignment-based tools cannot find. So they have a low number, but ProTrek can actually find those proteins have low TM scores. So you can see this. So this part shows that ProTrek can not only find a high similarity proteins, but also find proteins that dissimilar. So namely remote homologous proteins. So ProTrek do research based on embedding calculation. So this strategy enables ProTrek to perform more faster retrieval. So here we do the experiments. So here the time is the total cost of finishing 100 search against a UniRef 15 database. And you can see for ProTrek, it only takes around 9 seconds to finish the retrieval. And even for the fastest sequence-based tool, Diamond, it takes around 200 seconds. And for the Seek, it will take more than 1 second thousand to finish this retrieval. So this shows ProTrek can not only achieve accurate retrieval, but also perform very fast retrieval. We have provided a online server for free trial, and due to the hardware limitation and the communication delay, uh, the search time may be relatively long, but we are working hard to overcome these problems. Additionally, we also test the ProTrek's transfer learning. So we use the sequencing coder of ProTrek, and we fine-tune this sequencing coder in many downstream datasets, and we found that compared to ESM-2 or ProtST, ProTrek outperforms these models in many of the tasks. For example, the subcellular localization, the AAV task, or the structure class. So this means ProTrek has robust transfer learning capabilities. So that means ProTrek can also be used for property prediction after fine-tuning. Like ESM2 or ProtST for supervised fine-tuning. So here are the basic introduction of ProTrek. So if you are interested, please check our paper or our GitHub repo. So if you have any question about the code or the paper, welcome to email me. And if you have any question, please let me know. Thank you.
All right, I think we can get into the Q&A session. I think there are a couple questions in the chat. Maybe we can go through them first.
Okay, I see. So the first one is the dataset used for training was not clear. You mentioned SQL has, but the final dataset, how was the number? Oh, okay. I will return back to live. So, I say the Swiss port has 500,000 proteins. However, one protein from this web page, you can see, one protein, it has many properties and text description. For example, a protein, it has function description, it has protein name description, or subcellular localization. So these 15 million samples is a map from multi-proteins to multi-text. One protein, it can have 10 text description, and one text description can correspond to multiple proteins. So here, 15 million samples include the multiple protein to multiple protein mapping. So it's not just the number of proteins, but also the protein text pairs. Uh, one why 6 contrastive learning loss function if I mask? Okay. So, for example, for the sequence and the structure, given we input a batch to the model during training. So, for example, we have 100 samples, and so we have 100 protein sequences and we have 100 structures. For each sequence, we want the model to predict which one of the protein structures is the correct one. So here we have the cross entropy loss for protein sequence. Here is one contrastive learning loss function. And also we can do the same function for the structure to find the correct sequence. So for sequence structure, we have 2 loss functions. And also we can do structure text and text sequence. So overall we have 6 contrastive learning loss functions. So how many tokens were applied for this model? Could you please mention the total text size you applied LM for it? I want to get an estimate token required for your. Sorry, we don't calculate in the token level. Just this 50 million and this 40 million are the size of proteins. And if you want to know the tokens, sorry. Uh, if you want to know the size of tokens, so I remember the token, the average length of text is around 40 tokens. So we have 40 million tuples, and the tuple contains the over average text length is 40 tokens. So maybe you can just times them, just 40 million times 40. So for your retrieval experiments, the retrieval experiments are the function descriptions of the protein. Are the function descriptions based on experimental, or are they geo terms? So do you mean by this table, this figure, that rackage? If you mean by this figure, so
Right, yeah. Can
yes,
you,
okay, thank you.
yeah, so
So
what
this
thing was, right?
what
Can you go to the next figure?
next figure, this one?
Yeah, right. So so when you're comparing, right, so you have a ground truth data set. So you have picked up all the proteins which has this function, right?
Yes.
So when you are doing the search, right, like now when you're doing retrieval, so are these functions what you describe here for the proteins in the data set, are they based on some experimental evidence or are they assigned using Go terms
Uh,
for the ground truth as well?
they are geo terms, but this annotation, we use the test set are chosen from the SweetProt database. So they are human reviewed, so they are reliable. So here, this function is from geo terms, but they are also reliable because they are under human reviewed. So they are you can say they are based on experimental evidence.
Okay, so the ground truth data set is from your SwissProt data set.
Yes, they are
Okay,
all from SweetProt database.
and can you go to the next slide?
Next one, this one?
Yeah, so even in this, right, so you have like 25 million.
Yes,
These
yes.
these
Sure.
are I think unit of 50, right?
So what? Pardon?
This is UniProt 50, right? The database from which you're trying to retrieve.
No, no. This is the SwissProt database for experiments, not the UniProt 15 database.
Okay.
Okay. So one clarifying question. The UniProt knowledge consists of 2 sections. Section contain. Yes. that is the SwissPro database and the Tremble database. Do you extract the manually? Do you extract the manually verified function only, or also Tremble? Have you ever benchmarked if manually verified function do better or the same as? Yes, actually, we extract the functions from both SwissPro and Tremble database, and we also know that the annotations from the Tremble database are not reliable. So we first used the annotations from we first use these 15 million. We use these 15 million samples to train the first version of ProTrek, and then we use ProTrek to filter out samples from Tremble. And these 25 million samples are relatively high quality because it went through the filtering. So actually we did the experiments to test whether we only need to train models on SwissPro database. We pictured it in our appendix. Maybe I can show you our paper in the appendix. Sorry, I can switch the screen. Please wait a second. Okay. Hello, can you see the paper?
Yes, we can see the paper right now.
Okay. So here, let me check. Yes, this one. So here we test the ProTrek trained on both UniProt 50 and SwissPro, or ProTrek only trained on SwissPro. So you can see overall the model trained on both UniProt 50 and SwissPro obtained higher performance than ProTrek only trained on SwissPro. So that is why we use both SwissPro and UniProt 50 samples to train our model. Also, some annotations are not accurate, but we use model to previously filter out, and the remain 25 million samples. Also, they also can contain some noise, but it can still improve the model's capability. Maybe I can.
Hi, can I ask a follow-up question?
Yeah, okay.
Yeah, so there are two questions I wanted to ask. So one is like, where you're saying you're filtering the UniRef dataset, right?
Yes.
So do you filter it based on some score, or like how do you kind of use your initial model to filter the UniRef dataset?
Sorry, do you mean that, so how we filter out samples from UniProt 50 database?
Yeah, based on your initial ProTrek model, which was trained on SwissProt. So how do
Okay.
you use that model to filter UniRef?
Yes, we do not do many investigation to test a different filtering strategy. So we use a heuristic empirical rule. So when we have a ProTrek model and we compute the ProTrek score, so ProTrek can do retrieval because it can give a score for a protein text pair. So we calculate all protein text pairs in the UniProt 50 sample database, and we set a threshold for these samples. So if the protein text scores are higher than this threshold, we keep them. Otherwise, we filter them. So this threshold is determined by averaging all the scores computed in the Swiss prot samples. So we have a ProTrek score, and for all these 15 million samples, we compute them. We compute their protein test scores, and we average them. So by doing so, we get a threshold. And for all samples in the UniProt 50 database, we filter out all protein text pairs under this threshold. That's a heuristic rule.
So is it that you take the embeddings of your sequence and the text, and then you use
Yes,
the cosine similarity metric to
yes,
find
yes,
the-
yes.
Okay.
对, yes, our ProTrek actually learns a joint embedding space. It can encode the text, sequence, and structure into a joint embedding space. That's why it can support snide retrieval tasks, because all points are in the same embedding space, and we compute their similarity based on cosine similarity, and also it will be divided into we will divide it by temperature. Yes.
Mhm. Okay. So the other question I have is, like, I didn't understand.
Sorry, can I interrupt just to give others a chance?
Yeah,
Sebastian,
sure.
sorry.
Sorry.
So there are some
Okay.
questions on fine tuning. If you don't mind answering that, Chin. So can fine tuning be done just on the sequence, or do you need all three tracks to do the fine tuning?
Fine tuning. Do you mean by this figure?
Yes. Mm-hmm.
Fine tuning. Okay. So this fine tuning, we follow the normal paradigm. So just we only use the sequence encoder, because the sequence encoder here is the backbone of ESM-2. So by extracting the sequence encoder, it can be fine-tuned normally, like other models, like so ESM-2 or other like separated SA prot. So just we input a sequence and a label, and we fine-tune them, and we test its accuracy. So this process doesn't include the text or structure, just a normal fine-tuning.
Okay, thank you.
Okay. And I can see what would be the best approach to search for homologous using ProTrek. Given the existing enzyme, would you recommend using sequence function or structure first to or you can use all of them simultaneously? Maybe. Yes. So actually, you can use all of them simultaneously. However, so in our empirical study, we found that usually using sequence for retrieval would achieve the best performance, because here the sequence encoder is contains 650 million parameters. It's a ESM2 backbone, and also this structure encoder is also a ESM2 backbone. However, it only contains 150 million parameters. Also, because here we encode the protein structure using Foldseek, and the Foldseek sometimes may not that accurate. So if you input protein structure for retrieval, the inaccuracy of alpha fold structure and the Foldseek may limit its accuracy during retrieval. So here we mostly recommend you recommend you use sequence encoder and text encoder for search. You can, for example, you can input a protein sequence and find their nearest sequences, or given a sequence you predict its or given a desired function, you find this function's nearest proteins. So both sequence to sequence or text to sequence is okay, and the structure to sequence is also okay. But if you want to get more comprehensive results, we recommend you simultaneously use all of them, and you can do further analysis to decide which one is better. So here, ProTrek outperforms ESM2 in 9 of 11 what characteristics in the remaining two tasks made ESM2 perform better? So I can see, actually ESM2 performs better in the stability and binding set detection tasks. So, you know, generally we cannot obtain a totally foundation model that can outperform all other models in all tasks. It's very hard. So I don't know. So I don't know. There is one model that can say it can beat every model in all tasks. So it's very hard. So during the training, the protein can learn the representative information of protein sequences, but it can still be biased by some patterns. So I think this would lead them would lead them to not perform well in all tasks. It's very hard. So I cannot tell. So on Earth, what factors impact the final performance? But I just can say it's very hard to perform better in all tasks. Yes. Follow up question to Swissport data because you're matching one protein to multiple texts or vice versa. Is the data not redundant and can the model be overfit due to this? Yes, that's a good question. Actually, we before we pre-training our model, we did the preliminary investigation to for the text redundancy. Oh yeah, you can see our paper in this. We have listed a table to describe this data redundancy. So you can see the here. So this table, it might be hard to see. If you see this, so this Swiss port, so this is the data information in the Swiss port. So you can see we listed all subsections and their unique numbers and they are all numbers. Here num8 means the unique numbers of texts, and the num-a is the redundant numbers. So because you know a text can be mapped to multiple proteins. So we use this to calculate the sequence the data redundancy. So in our experiments we found that if you train with Swissport, it may not overfit because the text diversity in Swissport is enough. But if you train only with UniRef50 data, it can overfit because you can see here for example, you can see this, the function subsection. Can you see this? For the function subsection, we only contain 4,000 unique functions, but we get overall 50,000. We get overall 500,000 function descriptions, and only 4,000 are unique. Protein and text descriptions are unique. So that means if we only train ProTrek on UniRef50 data, it will overfit. That's why we have to combine both UniRef50 and the Swissport to train. In my opinion, the UniRef50 samples are like the synthetic data, and it cannot be directly used. It have to be combined with real data to avoid overfitting. So I might miss this, but we only fine tuning the sequence encoder impair the correlations between the sequence encoder and the other two. Oh yes. So here we don't mean you fine-tune model on this dataset and after that you do the retrieval. Actually, this is only about this is only to test the transfer learning capabilities. And if we fine-tune ProTrek sequence encoder in the downstream task datasets, we cannot use them for retrieval. So this is only for fine-tuning for the transfer learning test. Can you describe your accuracy metric for assessment? Yes, the MAP is from the information retrieval field. So the MAP, yeah. So in general, the MAP is about the overall retrieval accuracy. It not only considers the top retrieval candidates, but also the all retrieval. It focuses on the overall accuracy of the retrieval. So if you are interested in this, you can see our paper in the appendix. So here we detailedly illustrate the mean average precision, how we calculate this metric. If you are interested in this, please check our paper.
Okay, I think there's no more question. If there's anyone has any more questions, you can either unmute yourself, ask now. Okay, there's one more question, actually.
Okay, I see. Do the structures used for pre-training contain both PDB and AlphaFold DB? That's true, yes. So you can see here. So because we cannot only use PDB database because PDB only contains around 200,000 proteins. But we have 40 million tuples, and actually in our pre-training we contains around we include around 10 million protein structures for pre-training, and these protein structures were all downloaded from AlphaFold, and for every protein we have its UniProt ID, and we download its AlphaFold database structure from based on the UniProt ID, and then we constructed this sequence structure function tuples. They are all from AlphaFold database.
All right.
Yeah.
So let's thank Jing for this amazing talk today. It's really, really amazing and very lively discussion we have. And thank you everyone for joining us today. And the next talk will be on December 12th, which is also likely going to be the last talk of this year. And we'll have the protein generator team from the Baker Lab, and you can check the schedule on our website as well. And we will see you then on December 12th. And thank you very much, everyone.
Thank you.